In this session, we will talk about the roles and functions of sugars in biology. Back to our brain teaser from the previous lecture. So, can you have a 2-carbon monosaccharide as shown in the picture? So, what is your answer? Well, the answer is yes and no. Yes, because this sugar is known as glycoaldehyde and it is found in the dust and gas around a young sunlight star. While the answer is no at the same time, because biochemically speaking, a monosaccharide should consist of at least 2 hydroxyl groups. We know that sugars are sweet and soluble carbohydrates, but have you wondered why they are sweet? Now, it is because of the presence of hydroxyl groups. They are arranged in a particular orientation which can stimulate our taste receptor. So not everything that comes with the hydroxyl groups are sweet, such as alcohol, because you will need a particular orientation so that it can stimulate our receptor in a specific way. And now we'll proceed with the first learning outcome of this lecture, which is the roles of sugar acting as energy storage molecules. We all know that sugar or glucose is for energy. This is exactly why glucose is the principal sugar found in our bloodstream. Some cells, especially the brain neurons, consume glucose as the only source of energy, except during starvation, it may utilize ketone bodies as an alternate source of energy. But think about this, about 20% of our glucose go into the brain, which is quite a lot, given that the weight of the brain is only about 2% of our total body weight. So, what will happen if we store too many glucose in our body? First of all, it is going to cause diabetes, which can impair our organ function, such as kidney failure, and even worse, it can lead to an increased osmolarity within the cells, leading to cell lysis. So, what is the solution? Well, why not storing the soluble glucose in the form of insoluble polymers, such as starch and glycogen? Because they are insoluble, they are not going to increase the osmolarity, and therefore there will be no cell lysis. This is what exactly we observe in nature. As a result, you have polysaccharide acting as the energy storage molecules. So in plants, starch is the energy storage molecule. So showing here is the unbranched polymer of alpha-D glucose, which is known as amylose, and it is linked together by this alpha-1,4 glycosidic bond. Another variant of starch is known as amylopectin, which is a branched polymer of alpha-D glucose. So in this picture, you can see that in the main chain, it is linked together by the 1,4 glycosidic bond, but for the side chains, they are linked together by the 1,6 alpha glycosidic bond. Potato, for example, is an important source of starch. If you slice a potato, stand it accordingly, and put it under a light microscope, you are going to see these starch granules as the major organelles. And within these granules, you have amylose, which is the non-branch molecule, and amylopectin, which is the branch starch molecule. In animals, you have glycogen, which is also an energy storage molecule. However, glycogen can be very large, which is up to 120,000 glucose residues. Like starch, glycogen are linked by the alpha-1,4 glycosidic bonds for the main chains, while the branches are formed by the alpha-1,6 glycosidic bonds. Now, one special thing about glycogen is that it allows rapid release of glucose from glycogen because it is highly branched. Now, let me take you to one real scenario. When a cheetah is sprinting towards a gazelle from stationary, it only takes about 3 seconds to achieve the speed of 100 km per hour, which is faster than many race cars. And this also means that a lot of energy must be released at a very short amount of time. And that is only possible with the use of glycogen. Okay, in this diagram, I just want to show you how a highly branched glycogen molecule looks like. So you can see that there are so many branches, and the digestion of this single molecule can release many glucose residues at the same time. And in sports, there is a strategy known as carbohydrate loading, which is a strategy to boost glycogen levels prior to a sport event, and this can improve your performance. So here is how the effect of carbo loading on glycogen levels. The whole procedure will take about 7 days. In the first 3 to 4 days, one will try to reduce their carbohydrate intake while increasing their exercise intensity. So that will deplete all the glycogen stored in the muscles. While in the last 3 to 4 days, that person will increase the intake of carbohydrate while minimizing their exercise intensity. With that, one person can achieve the maximum level of glycogen within the muscles. Well, the major site of glycogen storage is our liver and the skeletal muscles. So here, for example, you can see that glycogen are stored within the granules in the liver cell and they are stained in pink. So here is another brain teaser from the previous lecture. Is polysaccharide synthesis an anagonic reaction or an exergonic reaction, and why? Well, the answer is A, an anagonic reaction. And that is because the synthesis of polysaccharide will require the input of energy, so it is energy demanding. And it is also an anabolic reaction, which involves the building of complex molecules from simpler subunits. On the other hand, the breaking down of glycogen to glucose or the breaking down of starch into glucose is an exergonic reaction because this reaction will release energy, and therefore it is a catabolic reaction which involves the hydrolysis of complex molecules back into their respective building blocks.